La profesora Luisa Serpel se graduó con honores de la carrera de Bioquímica y Genética en la Universidad de Nottingham y tiene un doctorado en Biofísica por la Universidad de Oxford. Actualmente es profesora de Bioquímica en la Escuela de Ciencias de la Vida de la Universidad de Sussex. Ella ha investigado por más de 25 años el mecanismo de autoensamblado de los péptidos y proteínas, y en particular en forma de fibra amiloide. Su investigación ha sido financiada por diferentes agencias que financian la investigación en Inglaterra, incluyendo Wellcome Trust y otras organizaciones que están más enfocadas hacia la investigación en Alzheimer. Ha publicado más de 110 artículos revisados por pares y revistas de alto impacto, y debe decirse que su grupo de investigación ha hecho una contribución significativa a la comprensión de la estructura genérica de las fibras amiloides, generando, es decir, publicando el primer modelo molecular de amiloide. En trabajos recientes, su grupo ha generado avances importantes en la comprensión de los efectos del péptido A-beta del Alzheimer en las células neuronales y las membranas celulares, permitiendo establecer vínculos entre el proceso de autoensamblado del péptido, es decir, la formación del amiloide, y su efecto tóxico en las células. Pues, sin más, dejo a la profesora Serpel. Ustedes.
Thank you very much, Luis, and all of you for coming. It's really a great pleasure to be here. I've had an amazing week. It's been amazing to see Mexico, and I really feel very privileged to be able to talk to you today. So I tend to speak very fast. So if I start to speak very quickly, please wave at me and say slow down, because I can't help it. So I'll try to speak slowly. So what I'm going to talk about today is the work that we've been doing to look at protein self-assembly. And the model that we particularly use, and the one that we're particularly interested in, is Alzheimer's disease. So I will start off by talking about Alzheimer's disease and our work there. And then later on, I'll talk more about the structural biology we've used to understand the assembly process and the structure of the amyloid fibrils. So just to give an introduction, as many of you will know, amyloid fibrils are well known because they are pathogenic. And I don't know if you can see here, this is an electron micrograph showing amyloid fibrils made from many different proteins. All of the electron micrographs you get from amyloid fibrils are very similar. So here we have Alzheimer's disease, in which the protein that forms amyloid fibrils is called amyloid beta. And I will talk more about amyloid beta in a little while. We also have a protein called transthyretin that misfolds in diseases called familial amyloidotic polyneuropathy, a number of other diseases, and this one at the bottom, which is famous because of Dr. Del Pozo Yauna, the AL amyloidosis, where we have immunoglobulin light chains that form amyloid fibrils. What's really interesting is these proteins are all very different from one another. They don't share sequence, and they don't share any structural similarities. And yet, they can all form these amyloid fibrils that look very much the same. So, these amyloid fibrils can accumulate in the tissues, and this is an example of an amyloidosis in which the A-alpha fibrinogen chain, a variant protein, forms amyloid. And you can see the staining of the amyloid deposition in the spleen in this very dark pink. So there's a lot of amyloid deposition. This is an insoluble aggregate that accumulates in the tissues. And in amyloid light chain amyloidosis, or AL, we again see this very large accumulation of amyloid fibrils. These are in the heart muscle from a patient with AL compared to a normal individual. So you can see this huge deposition of these amyloid fibrils. And what this tells us is that these fibrils are really important in terms of the pathogenesis of the disease. And this becomes particularly important when I tell you about the most recent findings in Alzheimer's disease. So now I'm going to focus on our work on the causes of Alzheimer's disease. This slide shows the model that we use. These are neurons, and then underneath are the astrocytes, shown in red. So the pathological hallmarks of Alzheimer's disease are two things. On the left-hand side, sorry, it's fallen off the bottom. On the left-hand side are the amyloid plaques. These amyloid plaques you can see in the electron microscope, and they have these fibers that are formed by a protein, which unfortunately is off the bottom here, is amyloid beta. They form extracellularly, so outside of the cells, and they cause these dense deposits, and you can see these very large aggregates within the brain tissue. On the right-hand side, we have neurofibrillary tangles, and you can see these are inside the neurons. They're in the cell body of the neurons. They form these large, dense accumulations of protein, and the protein is, underneath here, is tau. Tau is a protein that usually stabilizes microtubules. It also forms these amyloid-like fibrils. It also forms these accumulations, these aggregates within the tissue, but these are intracellular. In my group, we have focused on this protein, in particular, the amyloid beta protein. So to put this in some context, in the context of the disease, this on the left-hand side is a healthy brain, and on the right-hand side is from a patient with advanced Alzheimer's disease. You can see the huge degeneration of the brain tissue and these large vacuoles that develop. Within this brain tissue, we see these amyloid plaques. So this is a histological section. You can see these amyloid plaques are stained in yellow, very densely structured protein. Within this amyloid plaque are these amyloid fibrils. So this is an electron micrograph showing these fibrils, and these are actually synthetic fibrils we made in vitro, and you can see they're very, very long, unbranching fibers. They're around 100 angstroms in diameter, so very, very tiny, but incredibly stable and strong. And what's really interesting is that within these fibrils, we have a very specific structure. This is known as the cross-beta structure, and I'll explain a little bit more about this in a little while. But in this structure, the beta strands, the protein converts into a beta sheet structure, and the beta strands run perpendicular to the fiber axis. And what this does is it creates a very, very strong structure, hydrogen bonded all the way along the length of the fiber axis. The other thing is that it's this very, very repetitive structure that allows different proteins to access this very repetitive structure. So this is the structure of amyloid beta. It's only short, it's 42 residues long, and so it can fold up to form this beta bend hairpin that then can access this repetitive structure. If you take something like immunoglobulin light chain, which is obviously much larger, all you would need to do is to fold this more times to access this repetitive structure. So it can accommodate many different proteins. So one of the things that we're very interested in is why is amyloid beta toxic? How does it cause Alzheimer's disease? How is it involved in the neurodegeneration you see in patients? And what has been noticed in the last probably 15 years is that people have discovered, so researchers have discovered, that it isn't necessarily these amyloid plaques that are important in Alzheimer's disease. What people believe now is that these oligomeric species, these smaller species, are really, really important in causing the neurodegeneration that we see in Alzheimer's disease. So when we have amyloid assembly, we start off with a monomer, the A-beta peptide, which starts to form an aggregate. It then forms what we call a small oligomer, and I don't know if you can see, this is an electron micrograph. These are just small spherical structures. These start to elongate to form protofibrils, which then form amyloid fibrils and eventually a cluster or clump of fibers that we call the amyloid plaque. And what we want to investigate, what is it about these structures that can cause neuronal cells to become dysfunctional or to die? And so we have a number of ideas about this. So this is a very simple diagram showing the structure of a neuron. Here's the nucleus. We have mitochondria, lysosomes. Obviously, in a neuron, you have an axon where you have to have transport of cargo, and then the communication between the neurons is accommodated by the synaptic connections. And there are many ideas about how A beta might be affecting these cells, and each one of these processes appears to be very important. So it seems that amyloid beta has an effect on all of these things. And what is it that is similar between all of these organelles is the fact that they are all surrounded by membrane. And so the suggestion has been that the amyloid beta is able to permeate membranes. So it's able to enter the lysosomes and affect the lysosomal membrane. It's able to affect the mitochondrial membrane, the plasma membrane, and the transport of cargo, and to affect the synaptic vesicles within the synapse. So we have tried to investigate this in more detail, and this is some work that we did some time ago, where we took this oligomeric A beta and we added it to neuroblastoma cells. So these are SHSY5Y cells. So they're neuroblastoma, they're not neurons. The A beta is shown in red here, and what we see over time is that the A beta associates with the outside of the cell. So the cell is shown with the nucleus in blue, the cytoplasm in green, and the A beta in red. And what you see by 1 hour is association with the membrane, and then later on you see internalization of the A beta into the cells. And then we used electron microscopy to look in more detail at higher magnification at these structures. And again, this is hard to see, I think, in this slide, but we use gold labels that label the A beta specifically, alongside, which is off the bottom again here, sorry, an antibody against clathrin. So clathrin is the protein that is involved in endocytosis. And what we found here was that the A beta and the clathrin were found within the same vesicles at 1 hour. So we see internalization through an endocytotic mechanism. After 24 hours, we see A beta accumulating in these large vacuoles within the cell, and these label also with cathepsin D. And cathepsin D is a marker for lysosomes. So what we believe these are autophagosomes. These have been enlarged lysosomes where the A beta is not being degraded. So it's accumulated within these cells, which eventually could cause cell death. So I have a little movie here that shows the neuroblastomas. So the neuroblastomas you can see here, they're obviously alive, they're moving, and this is over 24 hours. What you start to see is some orange marker for the A beta, hopefully, within the cells that you start to see accumulating around here. So as the cells are being incubated with the A beta starts to internalize into these neurons. It's hard to see because they're moving a lot, but what we see is that they start to accumulate within here, and possibly, if you're watching this carefully, you can see some of the cells start to die. So they apoptose and die. So in this final one, you can see the A beta has accumulated within the cytoplasm, and probably in these small vesicles or vacuoles, which are probably lysosomes. So to look at the effect that this has had on the cells, we used electron microscopy again. So these are sections through the cells. And you can see here on the left-hand side an untreated cell where you can see the nucleus very clearly here and the nucleolus. If you're lucky, you might be able to see the mitochondria, and these are lysosomes. After we've treated the cells with A beta, what we see is that the nucleus already looks quite damaged. And this is the lysosome. You can see it's very, very different from this lysosome, where you've got this dense accumulation of protein within this lysosome. So what we see is that the A beta is able to cause degeneration of these neuronal-like cells. We also wanted to check that this also was the case for hippocampal neurons, so primary neurons, and again we found that the A beta accumulates within these neurons, and we are doing work at the moment to look at the effect that this A beta has on the synaptic connections between cells and the neuronal function. So one of the big questions in the Alzheimer's field is that all of us have A beta. Every single one of us has A beta, and the A beta, although we don't know what its function is, appears to be doing something useful. What is it that makes A beta start to form these aggregates, these oligomers, and these fibrils in patients who are suffering from Alzheimer's disease? So one of the questions that we asked was, is it possible that oxidative stress has an effect on A beta? And so this work was done by a postdoc in my lab, Yusra Al-Halili, and she looked at the fact that oxidative stress is implicated in triggering Alzheimer's disease and the importance of metals. So this is the A-beta peptide. And what you see is it has a tyrosine residue at position 10. And it's well known that tyrosine has the ability to form dityrosine, and that's shown here, in the presence of an oxidative environment, so in the presence of metals, and particularly in the presence of copper. So we wanted to investigate whether the formation of dityrosine caused by oxidative stress could be a trigger for the self-assembly of A-beta and the downstream toxicity. So first of all, she had a look at A-beta in vitro. So we know that we can form amyloid beta 42 fibrils in vitro, and then we incubated them in an oxidizing condition, which is copper and hydrogen peroxide. And this is a fluorescent spectroscopy which shows at 410 nanometers the increased intensity of a peak at 410, which is the dityrosine peak. We also checked this using LC-MS-MS, and we found exactly the same as the dityrosine standard. We also found this dityrosine within the oxidized A-beta fibrils that have been hydrolyzed to enable us to do the mass spec. We also then found that if we take A-beta monomer and we allow it to assemble in oxidizing conditions, again, we get the formation of dityrosine crosslinks. If you don't add copper and hydrogen peroxide, you don't get any dityrosine crosslinking. So this is not necessarily a spontaneous occurrence within A-beta. And then we did immunogold labeling using an antibody against dityrosine. And so you can see here these fibrils label nicely with the dityrosine antibody, whereas the control fibrils do not. So we can see that we've got dityrosine crosslinks within these fibrils. So one of the ideas is that this dityrosine is stabilizing these fibrils. They do look rather different from these ones that were formed under different conditions. So then the big question is this relevant to Alzheimer's disease? So we decided that we would have a look at some brain tissue from Alzheimer's patients. So this is an amyloid plaque, which I think I showed you earlier on. And so we used this tissue to see if we could find dityrosine crosslinks within this tissue. And I think this is going to be hard to see, so I apologize. So we're using gold particles, so immunogold labeling, that allows us to look for A-beta and also dityrosine. And so this is an amyloid plaque, and you can see it's very strongly labeled with the gold. And what you notice is that it has two different size gold labels, one for A-beta and the other one for dityrosine. And you can see that they are co-localized within this tissue. And to highlight this a little bit more clearly, the ones in red are dityrosine and the ones in blue are A-beta. So we see that there's dityrosine cross-linked A-beta within these amyloid plaques. Then we thought, well, if this is important in Alzheimer's disease, could this be something that we could use as a biomarker? Perhaps we could have, obviously we can't take tissue from patients, but could we have a look at cerebral spinal fluid from patients and see if there's any dityrosine cross-linked A-beta within the CSF? So we took some cerebral spinal fluid from Alzheimer's patients, and we used, again, immunogold labeling to see if we get dityrosine and A-beta labeling within this solution. So this is quite hard to see, but the large particles are dityrosine, and the smaller particles, which you can probably see a little bit more clearly up here, are A-beta. So within the CSF of a patient with Alzheimer's disease, we see both labeling, suggesting that we have dityrosine and we have A-beta. We can't be absolutely sure that it's dityrosine cross-linked A-beta because this is a solution. But although this doesn't look like a very compelling result, if we look at CSF taken from patients who do not have Alzheimer's disease, we see hardly any labeling at all. So this is an age-matched control, and there is no labeling at all. So we see very clearly that there's A-beta and there's dityrosine within the CSF of a patient with Alzheimer's disease. So we believe that the dityrosine formation of A-beta is really important. So what we think is happening is that the A-beta, in the presence of copper in an oxidizing environment, is able to interact and produce ROS, and starts to assemble to form these dityrosine cross-linked structures. And these can be internalized into cells, and I didn't show you this data because of limited time, but they can be internalized into cells and also are found within amyloid plaques and also in the CSF of Alzheimer's disease patients. So to summarize this part of my talk, the process of assembly from monomer to fibril produces these toxic assemblies. And I think it's really important, though, that the Alzheimer's field does not forget that amyloid fibrils cause mechanical damage. And if we take lessons from the other amyloidosis, we know for sure that these fibrils are really, really important. There's been a tendency in the Alzheimer's field to sort of sideline the amyloid fibrils and say these are not important, but I believe that they really are. So we found that the oligomers can enter cells, and this may be why they're more toxic, because they are small enough to be able to be internalized and cause damage within the lysosomal compartments. We know that A-beta also can induce the accumulation of autophagosomes, and normally we would not expect to see autophagosomes. They usually turn over very quickly. And so what this suggests is that the A-beta is causing an accumulation of autophagosomes, where the protein can't be degraded. I haven't been able to show you this piece of data, which I think is really nice. We used a model organism, which was a water snail, and we showed that A-beta alone can cause a memory loss. And so because I haven't had time to show this, I can very happily send you the paper or talk to you about it afterwards. And I've talked to you about the A-beta that can be stabilized by cross-links. So now I want to move on a little bit to the structure of amyloid fibrils. So the work that we've been doing over many, many years was really to try and understand what the structure of these fibrils is. And there are two really good reasons for doing this. The first is to try and understand why particular proteins have the propensity to form amyloid fibrils in disease. Why would they misfold? The other reason, potentially, is that these structures are incredibly strong and have a real potential for using as nanobiomaterials. So I will mention that a little bit in a moment. But we've been particularly interested in how the sequence of the protein determines whether it will form fibrils and what structure it then forms. So this is the other side of amyloid. Amyloid fibrils are not only pathogenic. In fact, they have recently been found in many different organisms. So, curly is an amyloid which is found on the surface of some bacteria, including Salmonella and E. coli. And what they do is that they protect the bacteria, and they also provide them with an adhesive property so that they can form biofilms. So these are the fibrils shown here. The reason I've shown this little spider here is because the spider, very interesting, is able to control the assembly of a protein. And these fibers that they spin into a web are very similar in their structure to amyloid fibrils. And what this tells us is that this organism has a very carefully controlled mechanism to prevent unwanted assembly. So we can learn a lot from these organisms that control the assembly very, very carefully. Whereas, of course, in the amyloidosis, we have many different proteins that have uncontrolled assembly, unwanted assembly. The yeast prions are well known because the amyloid nature of a protein called SUP35 or URI2P provides them with the possibility to have an inherited non-genetic element. And finally, this is in a human, so this is a protective amyloid. It's formed from PMEL17, and it forms this very protective layer around melanosomes in human beings. And these amyloid structures have not been that well studied yet. So what about the structure of fibrils? Well, I have to split it up into macromolecular and molecular structure. So on the left-hand side, I've shown some electron micrographs and some electron micrograph analysis that shows that insulin can form amyloid fibrils with many different numbers of protofilaments. So if you take an amyloidogenic protein and you put it in vitro and allow it to assemble, it will often form many different morphologies with perhaps 2 protofilaments or 6 protofilaments or something in between. What's really important, though, is that the structure within the protofilament is always the same. It's cross beta. This is some work that we did many years ago, 2000, where we did cryo-electron microscopy, and we were able to get very, very high resolution images of these fibrils. And we could see, if we zoom in, that there were striations along across the axis of the fibre, so perpendicular to the fibre axis, and this is the beta strands running perpendicular to the fibre axis. So they're only 4.7 angstroms apart, these striations. And then there are a number of structures that have been created and explained by solid-state NMR, where we can see, and this is A beta, where we can see the beta strands running perpendicular to the fibre axis with these two sheets. If you look down the fibre axis, you can see this very important contribution of the amino acid side chains. And this is a crystal structure, is the first of many, many crystal structures solved by David Eisenberg, where they have been able to take short peptides and show how the side chains of the amino acids interdigitate to form this steric zipper. So this is really important. Now, our method of choice has been X-ray fibre diffraction, and so I just want to explain that to you very briefly here. What we do is we make a high concentration solution of fibrils and we place it between two capillary tubes. The capillary tubes have got little wax plugs in the end, so that when you put the droplet between, it holds in place. And you can see in this schematic that the fibres are just distributed randomly in solution. As they dry down, they start to align and form a bundle of fibrils, and here we have a bundle of fibrils where we have alignment. And the aim is to get as good alignment as possible, to get as good diffraction as possible. So this image shows you a picture of what the fibre looks like. So it's only about 1 millimetre high. But you can see that under a cross-polarized microscope, it has birefringence, which seems to suggest that we have some ordering of the fibres within this structure. So we can put this into an ordinary X-ray crystallographic diffractometer and collect a diffraction pattern. And generally from amyloid fibrils, we get this very specific cross-beta diffraction pattern. So I'm going to explain what this diffraction pattern means very briefly. This is the fibre axis, and so it's vertical. So we place the fibre vertical. We have a reflection at 4.7 angstroms. So I mentioned that earlier when we were talking about the cryo-EM striations, very, very close together. This is 4.7 angstroms, and then on this axis, which corresponds to everything across the fibre axis, we have a reasonably strong but quite diffuse reflection around 10 angstroms. So let's see where they come from. So this is the structure that I showed you earlier. So the 4.7 angstrom reflection comes from hydrogen bonding of these beta strands along the fibre axis. It's very, very strong and very intense because this only shows a small part of the fibre. It's much, much longer than this. So all of those structures contribute to this incredibly strong and intense reflection. If we look down the fibre axis, we have a spacing of 10 angstroms, and this corresponds to this 10 angstrom reflection on the equator. And this accommodates the side chains, which I will show you in the next slide. I thought that there was some here. So this is an array of diffraction patterns that we can get from amyloid fibrils. Depending on how much information we can get a lot more information about the structure. So for example, here we have a simple cross-beta diffraction pattern with just those reflections I mentioned. We can say that this is cross-beta. As we increase the ordering of the structure, we can get more information from the diffraction pattern, and we can start to think about how the peptide fits into a unit cell, which is right down here. This is a very highly oriented diffraction pattern, and the fibrils were really quite crystalline. And so the diffraction pattern gave a lot of information that allowed us to build a model of the structure. And you can see, this is looking down the fibre axis, and you can see the interaction of the side chains that form this steric zipper. And this, I'm going to mention a little bit more about this model system a little bit later on. This is actually an electron diffraction pattern. So it's taken in an electron microscope, and we can, again, build the structure of the fibril. I'm going to explain a bit more about how that's done in a moment. So the first project I want to tell you about used this algorithm. So this is an algorithm called Waltz. I said right at the beginning that there are many, many different proteins and peptides that can form amyloid fibrils, and they don't have any sequence similarity, nothing obvious. And so a number of different groups have built algorithms that can predict amyloid propensity. And they use lots and lots of different factors. This particular one can predict amyloid fibril formation from sequences, and it has a high sensitivity to less hydrophobic sequences. And it's position specific. So if I just try and explain this table very briefly, this is based on a 6-residue peptide, and where it's green, it suggests that this amino acid will be nicely accommodated within an amyloid fibril, and where it's red, or bright red, it's less accommodated within the fibril. And what you notice, for example, for lysine, is that lysine is nicely accommodated on the outside of the peptide, but not really on the inside. It's interesting that position 4 is quite happy to be there. And so they did exactly the same thing for each of the 20 amino acids to see, to be able to predict the best amyloidogenic sequences. So we know that we can form diverse fibrous structures formed by amyloidogenic peptides. I'm going to tell you a little bit more about each one of these peptides, not this one, and this one. So I'll talk to you a bit more about those in detail, but these 3 peptides were identified by Waltz. And so we were interested to see how this sequence, which is we call HIFNIF, but is histidine, tyrosine, phenylalanine, asparagine, isoleucine, phenylalanine, forms these beautiful fibrils in the electron microscope. And we can see that the structure is clearly quite different from this other 6-residue peptide and this 5-residue peptide. These provide us with really nice models to be able to really try and understand exactly how the sequence contributes to the structure of the fibril. We used a method called linear dichroism that really helps us to give orientational information about particularly the tyrosine residue. It would also work very nicely for tryptophan. So this is a method very similar to circular dichroism, except that the fibrils become aligned. So you get orientational information. So just to explain this very briefly, this 200 nanometer peak comes from this beta strand. This is the fiber axis. And then if we zoom in on this part of the graph, we can see the contribution of that tyrosine residue, which is shown here. So the tyrosine residue is contributing to the sheet spacing between the beta sheets. And we can use the fiber diffraction, the electron microscopy, and the linear dichroism to build models that fit the data. So these are the 3 fiber diffraction patterns that we get from these 3 peptides. And again, you can see that they give us this classical cross-beta diffraction pattern with a number of other reflections that we can use to build the models. So I'm not going to go into this in a lot of detail, but to show you that we were able to build models from each one of these fiber diffraction patterns. And the way that we test whether the models are correct or not is to calculate the diffraction patterns from these structures and then compare it to the experimental data. And so the idea is that we want to get a diffraction pattern that's calculated as close as possible to the experimental data to say that this is the best model that we could build that fits the data. So really, to summarize this, this is a model of the structure of, let me get that out of the way, of the HIFNIF peptide. So what you can see here is the beta strands running perpendicular to the fiber axis. Look at all those hydrogen bonds that produce this incredibly stable structure, telling you that this can be useful and also why the body can't degrade these. Here you can see, looking down the fiber axis, the side chains that interdigitate to form this steric zipper. So this is very, very tightly packed, and it tells you why the sequence of a peptide is really important for the propensity to form fibrils and the final structure. So to look into the contribution of side chains in a bit more detail, we used another model here, which was the peptide has the sequence KFFE, AAK, KFFE. I want to use this as an example for how you can change sequence to look at the change in structure, and also then to show you how we might use these fibrils in nanobiotematerials. This is a nice peptide because it forms these nanocrystals. In the electron micrographs you can see striations, and this is an atomic force micrograph. So you can see that it forms these crystals, and we get both X-ray and electron diffraction, which gives us enough information to be able to build a model. So this is looking down the fiber axis, and you can see those phenylalanine residues are really important in holding together the structure. And what we notice when we look at the sequences of amyloidogenic sequences is that they often have these aromatic residues. They often have a phenylalanine, a tyrosine, and often a tryptophan. So this forms pi stacking between the sheets and also along the sheet axis. So again, what we were able to do here was to take this diffraction pattern, calculate the most likely unit cell, model in the structure, and then calculate a diffraction pattern from that model structure. So again, you can see this is experimental, and this is a calculated diffraction pattern. And we wanted to know what drives fibril formation of this structure. And so what we did was we took each of the positions and changed them to different residues. And we didn't do it exhaustively. We just did it with a few of the peptide, of the amino acids. So we changed the lysines to either alanine or to arginine, and we tested the changing phenylalanine to alanine. And we found that if you take phenylalanine out, any of the phenylalanines, then you prevent fibril formation altogether. So those pi-pi stacking arrangements are really important. If you remove the charge by removing lysine and replacing it with alanine, what you find is that instead of getting the lateral association of the fibrils, you get only individual fibrils, thinner fibrils, and they're shown in this micrograph here. So phenylalanine plays a really essential role in assembly, and lysines influence the lateral association. So they seem to be on the edge of the fibrils and are causing that self-assembly not only in the fibril direction but also laterally. If we change the lysine to an arginine, which is a larger amino acid, we see that we change the sheet spacing from 10.66 angstrom to 11 angstrom, so slightly moving the sheets apart. So what this does is it gives us two things. One is to tell us that our interpretation, our prediction is correct for this model structure, and that you can manipulate the sheet spacing by changing the amino acids. So this is our example of a functional material that we can use. So we can take these peptides, and actually we've taken many of the different variants of this peptide. I'm just showing you one example here, which is where we've replaced the lysine for an arginine at position 9. Forms beautiful amyloid fibrils. If we incubate it in the presence of tetraethyl orthosilicate and then wash with ethanol and water, we end up with so first of all, just to say that these are indeed cross beta. And so we end up with fibers that are coated with a thin layer of silicon. So what we've made here is very, very, very narrow silicon nanowires. We found that if you do a diffraction pattern from these fibers, you still get the cross beta diffraction pattern, although it's very, very difficult to see, suggesting that the protein, the core, is still within these fibers. And then we can do we can embed these fibers and section them, and you can see that they have then an electron-lucent core, which is the protein, and then this much more densely staining silica coating. And if you occasionally we get a cross-section, so you can see these little ring-like structures. So we've got these silicon nanowires. And by looking at lots of different variants of the peptide, we found that some of the positions within the sequence are really important for the templating conditions, and so we found that particularly these edge lysines are really important to enable the silica templating to occur. So this is my final example of a amyloid structure, and the reason I like to show it is because it highlights the possibility that you can form something that's still cross beta, but it forms a very, very different morphology, and these are very beautiful, I think. So this is an octapeptide from alpha-synuclein, and it forms helical nanotubes. It only forms them at incredibly high concentration, so 180 mg per mil. So it forms these very thin-walled structures. And what you can see here from these electron micrographs is that sometimes these start to unravel a little bit like an old-fashioned telephone wire. So we were interested to see what these structures looked like. And so we were able to get nice diffraction patterns. And again, we've got 4.8 angstroms reflection on the equator, on the meridian, sorry, on the fiber axis, but also this more sort of 10 angstrom reflection also on the fiber axis. And I can explain to you a little bit more about why that occurs, but I think the best thing to do is just to explain how we interpreted this. So we found that we can build a classical cross-beta model to fit this data. So we have the beta strands running perpendicular to the fiber axis, the side chains interdigitating, and we can calculate the diffraction pattern that's shown here to show that it's very, very similar to the experimental data. But how do they form these helical nanotubes? Well, if you look at the sequence of the peptide, it's amphipathic. So it has a hydrophobic region shown in orange and a hydrophilic region shown in turquoise. And so what we believe is that the peptide orients itself into a sort of bilayer, so that it buries this hydrophobic region within this bilayer, and then it forms these helical structures that then can be enclosed to prevent the exposure to solvent. So it would explain why we sometimes see tubes and occasionally we see these unraveled structures. So really the reason I wanted to show you this was to show that we can form diverse structures by amyloidogenic peptides, and this might become very important, for example, in AL, where the immunoglobulin light chain is able to form amyloid fibrils, but also to deposit as crystals and other types of structures. So to summarize what I've talked to you about today, we can get a peptide to self-assemble to form amyloid fibrils. The sequence of the precursor seems to be very, very important to not only to decide whether or not something will form fibrils, but also to determine its eventual morphology. It forms this fibrillar architecture, and potentially these fibrils have some material properties. And one of the aims of this would be that if we know how a sequence will form a particular morphology, we might be able to design something that will form something specific. And we believe that elucidating what drives assembly is really important for understanding pathogenesis in a range of amyloid diseases, but also in terms of trying to understand how we might make new materials. So we want to dissect the assembly mechanisms and then solve the fibril structures. And in order to do this, we need a whole battery of different techniques that can be combined to give us the three-dimensional structure, the overall structure of the fibril. So we need to understand protein misfolding diseases and also to utilize this to actually use these peptides as nanomaterials. So I think I've just said this. So really, it just remains for me to acknowledge all the people. This is some of my research group on my birthday, and the people that weren't in that picture. All very happy. I've tried to mention or show a picture of each of the people that did the work, and a huge number of collaborators. Lewis is here, and we get funding from the Medical Research Council, BBSRC, which are both UK government research councils, as well as charities like Alzheimer's Research UK and the Alzheimer's Society. And Lewis has asked me several times whether Brighton, where I'm from, is on the beach, and this is the sea. So there's a pier, and although Sussex University is in Brighton, and the beach is actually full of little pebbles, so it's not sandy usually, but occasionally the tide goes out. This is all beautiful sand, and of course this is what England looks like all the time with this incredible blue sky. So thank you very much for listening.